In this lecture, we're going to provide further clarification to the definition of cancer. And we start with a couple of terms here. And the first term here is what we call neoplasia, where neo refers to new and plasia refers to growth. This is a process that is not only just normal in growing organisms, but this process can become pathological as it does in the case of cancer. And in the case of cancer, that process of new growth will eventually develop into what's called a neoplasm. And the neoplasm is a term that refers to the newly grown structure. And in this case, we're referring to a cancerous cell. Now, with this growth and development of new cells, division of cells, this process becomes uncoordinated and autonomous. Uncoordinated in the sense that there does not appear to be any sort of cellular regulation mechanism in that the cell will serve the function of the organ within which that cell is residing. And it becomes autonomous in that the cell appears to be not sensitive to various chemical signals inside and outside the cell, but the process of replication is just carrying out independent of anything, any other influence outside the cell. So that lack of normal, healthy regulation will result in an increased size of the cell and will also increase the number of those cells as those cells undergo division as well. One of the main regulatory mechanisms that maintains healthy tissue is inhibition. And so inhibition refers to physiological processes that basically puts the brakes on growth mechanisms so that cells and tissues do not overgrow. And one example of that, as you can see in this lower left-hand picture, what we're looking at here is the Belgian blue bull. And the Belgian blue bull is a bull that has a mutation in its own DNA profile. And that mutation affects a gene that will produce a protein, and that protein is illustrated here in this computer reconstruction of what's called myostatin. And myostatin is a protein also referred to as GDF8. GDF stands for growth and differentiation factor. And this particular protein is a negative regulator of muscle growth. And so a negative regulator of muscle growth does not necessarily stop a muscle from growing, but slows down and prevents the overgrowth of muscle. And an overall more detailed summary of this particular protein is outlined in this research paper, and you have the citation here. So what we see here with the Belgian blue bull, we see a bull that is abnormally heavily muscled because this bull has been bred to have a mutation in the gene that makes myostatin. And if the gene is mutated, the protein will be mutated. And a mutated protein will not be able to carry out the natural function of that protein, which is to put a regulation on muscle growth. That's what we see here with the bull, is that without the inhibition regulation, muscles grow uncontrollably, as we can see in this photo. We move on to just some basic definitions of cancer. One common term here that we'll get into and that you hear about when reading other sources on cancer is the tumor. And the tumor is a collection of cells that are growing beyond what is considered to be healthy or normal for those cells and that tissue. The term neoplasm um, is not one that is synonymous with tumor. So tumor is the most general description that we have here about a mass of cells, but there are two types of neoplasms. The first one here is referred to as a neoplasm that is benign. And being benign, this refers to these abnormal cells, and these abnormal cells are differentiated. And so these are cells that have developed that still possess characteristics for the tissues within which those cells reside. And these differentiated yet abnormal cells will cluster together into a single mass. And these cells tend to be isolated from other tissues. And there's usually a relatively a tough capsule that surrounds these cells inside the tissue that separates these unique cells, abnormal cells, from the healthy normal cells. Benign tumors tend to be easier to treat because these are tumors that can be much more easily isolated and targeted with surgical treatment or radiation treatment. The second type of neoplasm is what's called a malignant uh, uh, neoplasm. And these are cells that are less well differentiated. So if we go back to the benign type of neoplasm, here we see cells that have all the characteristics of liver cells inside the liver, but are abnormal enough where they are clustered together. In the malignant tumor or the malignant neoplasm, these are cells that are not well differentiated. And the cells that are not as differentiated does have the potential and will leave the primary tissue and can enter the lymphatic system or the cardiovascular system. And the problem with a malignant tumor or a malignant neoplasm is that these cells, when entering the lymph or cardiovascular system, can travel throughout the body and can find basically a landing point in other tissue. And in doing so, these cells, when entering other tissues, can form secondary tumor sites in those other tissues. So it's not uncommon to find a cell that has some of the features, not very well differentiated, but some of the features of a liver cell embedded in other tissues because those abnormal cells found a way through the cardiovascular system or the lymphatic system to travel to another part of the body. To understand the pathophysiology of cancer, it helps to begin with understanding what normal tissue renewal and repair is all about. Proliferation is a normal process of cell division, and it's an adaptive mechanism based upon the stresses placed upon cells. And so this normal process functions to replace old cells or dying cells. And two examples here include the skin and the intestine. The basic type of cell that makes up the outer layer of the skin and the inner layer or the inner surface of the intestines are epithelial cells. And these are cells that have relatively rapid rates of turnover. So once a cell is born, it has a very short lifespan and is replaced. And this is a normal process for the skin and the intestinal system. Differentiation is where cells become more specialized with each time that cell divides. And so cells will differentiate into a white blood cell, for example, or a red blood cell. Now, these are two clearly different types of cells found in the cardiovascular system, yet both of these cells have the same birth from stem cells. So stem cells are the most undifferentiated type of cell that can develop into any other type of cell in the body. And then once again here, this is a normal process that takes place. Apoptosis is a term that refers to, and I'll put this in quotations, pre-programmed cell death. What this basically means is that every cell in the body, to varying extents depending upon the type of cell, will have a very definitive lifespan. And that is the normal process of tissue and cellular turnover that takes place in the human organism. Why this is a normal process is because during cell division and cell specialization, cells do become damaged. And so the copying of and passing on of DNA through cell division is a nearly perfect process, but sometimes errors result during the normal course of cell division. And when that happens, DNA damaged cells runs the risk of developing that cell into a cancerous type of cell. So one of the safeguards that nature put into these cells is apoptosis. So proteins made by that cell will ensure that that cell has a definitive lifespan. And so in a DNA damaged cell in a healthy organ, that cell would normally have a much shortened lifespan before it has the chance of growing uncontrollably and dividing uncontrollably. Apoptosis will also serve to eliminate old cells or senescent cells. So as cells age, those cells reduce the capacity for carrying out the basic functions of the cell to support the organ inside that tissue and that organ itself. And so having that built-in mechanism where proteins will begin the process of ending the life of that cell will ensure that new cells will take place of those older cells and support the function of that tissue and that organ. So this ends our second lecture on cancer definitions.